in chapter 4. Now let's begin the session. First, let's see the structure of typical plant cell. And this we have plasma membrane. Then in the internal content of the cell we have the nucleus, which is very prominent and which is controlling all the other actions of the cell. Then we have endoplasmic reticulum, mitochondria, Golgi apparatus, chloroplast and the big sized vacuole. Now children, we can see clearly this plasma membrane is separating the external environment and the internal contents of the cell. Now activity 1, what gets in and go out? Some substances are used by the cells while some are discarded by the cells. Identify and tick mark which substances should go in and which substances should go out of the cell. Which substances should enter the cell and why? Which substances should go out of the cell and why? Now let's see the table. Substance should go into the cell, should go out of the cell. Now first substance is oxygen, glucose, proteins, fats, vitamins, minerals. These are all the substances which are utilized by the metabolic activities. Now these substances should go into the cell. Now carbon dioxide and the waste should go out of the cell. Why waste substances are supposed to go out of the cell? Because if the waste remains in the cells, that will become toxic to the cell. So at the regular intervals, the waste substances should go out of the cell. Next, can you name any other substances which should enter into the cell and which substances should enter into the cell and go out of the cell? Next, preparing solutions, preparation of saturated solution, and which one is more concentrated solution? Children, let's see all these three activities in one video. So let's begin the video.
Hello children, welcome to the other session. Today we are going to deal about the topic plasma membrane, chapter 4 in 9th standard. So let's begin the session. Children, let's prepare solutions. For that we need water, sugar or salt. Now water is the solvent, sugar or salt is the solute. Now the solute, that means sugar or salt, must dissolve in the water. That will make the solution. So let's begin the first activity, preparation of saturated sugar solution. For that we need water, sugar, beaker. I have filled water in the beaker. Now I'll add some amount of sugar. Stir it well. Repeat it until some amount of it is left and dissolved. So this is saturated sugar solution. Children, let's see which one is more concentrated. For this we need 3 beakers filled with water, sugar. Now I am adding half spoon of sugar in first beaker, 1 full spoon of sugar in second beaker, and 1 and solute is completely dissolved in the solvent. Now children, beaker 1, 2, 3. Which one is more sugary? Yes, the third beaker. Why is that? Because I have added 1½ spoon of sugar in the third beaker. Right? What is the difference between first and third beaker? In the first beaker I have added half spoon of sugar. In the third beaker I have added 1½ What can I do to make the first beaker sugary? I have to add 1 spoon of sugar in this beaker. So this is the concentration of solution. The third beaker is more because of the sugar present in it.
Now children, you have seen how we have prepared solution, sugar solution, and which solution has more concentration. Now let's observe observation of material in different solution. First, observation of dry kishmish in water, then observation of swollen kishmish in sugar solution. So let's see the video.
Children, let's observe the material in different solution. For that, I am taking 100 ml of beaker and I am adding water. Then I am adding 2 raisins, that is dry kishmish. After 1 hour, let's observe what is happening. You can see children, the kishmish swelled. That means the water is getting inside the kishmish. Now add some sugar and water. Now children, I have prepared saturated sugar solution, mixing sugar with the water. Now I am taking one of the swollen kishmish and adding to the saturated sugar solution. I am leaving this experiment for an overnight. Children, you can see the swollen kishmish which I dropped in the sugar solution is shrinked. Right? Let me show you clearly. Shrinked kishmish.
So children, we have seen the two activities. In the above two activities, the cells in the outer layer of the kishmish permit to pass through, right? Next, to prove osmosis using potato. For that, materials needed are 1 raw potato, 1 boiled potato, 1 bowl, 2 pens, water, sharp knife and saturated sugar solution. Let's see the activity by using the video.
Children, let's do some activity to observe the process of osmosis in the potato cup. Now pour sugar solution into the trough. Let's make the potato cup. Potato and a knife. Peel off the outer layer of the potato. Now the potato cup is ready. Now keep the potato cup in the trough which is filled with the sugar solution. Now we need to add some more sugar solution to the height of the half of the potato cup. Yes, half. Now take some water and pour it in the potato cup. Now pierce the pin to know the level of the water in the potato cup. Children, observe very carefully. We have added sugar solution in the water trough and water in the potato cup. I have marked the level of water by piercing a pin. Leave this experiment for half an hour. Now children, I have made another potato cup. Placing this potato cup in the trough which is filled with the water. Now I need to ensure that the height of the water should be half of the potato cup. Yes children, now I need to fill sugar solution in the potato cup. Now I am marking the level of sugar solution by piercing a pin. Now children, we have two different bowls. In one bowl I have added sugar solution and water in the potato cup. Here, now in the bowl I have added water in the bowl and sugar solution in the potato cup. So in the potato cup experiment, let's see the observation. Children, you can clearly see the water in the potato cup has moved towards the sugar solution. See, this was the pin which I pierced to see the level of the water. Now there is no water left. That means the water in the potato cup has moved towards the sugar solution. Now coming to the second bowl. Now the bowl was filled with water and potato cup was filled with sugar solution. Now the mark which I pierced with the pin, the water level has raised in the potato cup because this potato cup has sugar solution. Right children? So we can clearly see by this experiment the water is moving towards the highly concentrated solution.
So children, let's see the result. By the above activities, we can say that the water always moved towards the sugar solution. That means that water is moving from the lower concentration to higher concentration, right children? So such process in which the water moves towards the sugar solution is called as osmosis. So in Greek, osmosis means pushing. We can see that the movement of water through the membrane in which the osmosis took place from lower concentration to higher concentration. So children, let's do the other activity to prove the process of filtration. For that, the materials needed are 2 beakers, funnel, retort stand, iodine, wheat or rice flour or cotton cloth. So let's see the activity by playing the video.
Add some wheat flour to the beaker and mix it with the water. I have added iodine solution to the solution. Now mix it and filtration. Now children, you can see the dissolved substances could pass through this cotton cloth. The undissolved substances could not pass and remained on the cotton cloth. So the materials which are dissolving in the solution is getting down and the substances which are not dissolving are remaining in the cotton cloth.
So children, by all the activities we have seen, the water is moving towards the lower concentration to higher concentration. What is the higher concentration here, children? Yes, I made a sugar solution, right? How did I make a sugar solution, children? I took some water and mixed with the sugar. That is what I made the sugar solution. So let's see the nature of the plasma membrane. It allows water and certain materials dissolved in water to pass through it. It will not allow certain substances or materials to pass through it. Allowing materials to pass through is called as permeability. Impermeable. Children, impermeable means it is not allowing any substances to pass through the membrane. Next, permeable. Children, permeable means the solute plus solvent is making solution, right? So what is the solute here, children? Sugar or salt. So the salt or sugar are mixing with the water. That is making the solution. Now the solute has mixed with the solvent, so it is permeable, right children? Semi-permeable. See, you can clearly see the semi-permeable is the membrane in which the molecules are entering from lower concentration to higher concentration. Next, selectively permeable. Only certain selective permeable things are allowed here. You can see there are certain substances here. This is extracellular fluid. This is the channel protein, this is the plasma membrane, and this is the cytoplasm. You can clearly see that the materials are entering it. Not all the materials are entering, children, only the selective permeable substances are entering. So plasma membrane is a selectively permeable membrane and living flexible membrane. So what are the functions of plasma membrane? It gives shape and specificity to the cell. Then mechanical barrier. See children, mechanical barrier. This is the wall which is called as plasma membrane. Assume my hand as the plasma membrane. Now this plasma membrane is stopping or hindering or not allowing all the substances to get into the cell. Just imagine this as the external environment and this as the internal content of the cell. Now this is the plasma membrane. It is not allowing any substances which are harmful for the cell. What are the internal content of the cell, children? Yes, we have nucleus, we have cytoplasm, we have all the cell organelles, that is endoplasmic reticulum, the Golgi apparatus, the lysosomes, isn't it children? So these are the cell substances which are not supposed to get disturbed. What is the function of the plasma membrane? Yes, it is acting like a mechanical barrier. Now, selectively permeability. We have seen in the process of filtration, children. What did I do in the filtration process activity, children? Yes, have mixed wheat flour with the water and iodine it. Now, iodine is easily soluble in the solution, but some amount of the wheat flour is not soluble, so it remained on the filter paper, right? So that is selectively permeability. Next, recognition. Recognition. Children, this plasma membrane plays a major role in recognizing the different types of the cells. Why? What is needed? Because it has to see whether any foreign particles are getting into it, isn't it? Because if the foreign particles get into the cell, it may disturb the internal contents of the cell, which will disturb the cell, which will lead ultimate death of the cell. So it has specific centers for recognition, isn't it? You can see clearly in this, receptors are present. With the help of the receptors, these are able to bind. Next, endocytosis. Children, this is seen in amoeba. You can see clearly the substances is getting closer to the amoeba. Next, it gets dissolved. Next, it gets completely dissolved in the amoeba. This is called as endocytosis. Flow of information. Children, you can see the water soluble molecules are entering to the plasma membrane, right? Then cell continuity. Why there must be cell continuity, children? Because the cell must form a plasma desmata. Plasma desmata. cell adjacent to another cell, cell adjacent to another cell. This forms a cell continuity. So the flow of information is taking place through the plasma membrane. Then specialization. Children, in the small intestine, this plasma membrane plays a major role. You can see the finger-like projections, isn't it? Villi. In the small intestine, this plasma membrane plays a major role in absorption of proteins, isn't it? Right. You can see the microvilli, right children? This is the crypts. This is the villi. I told you the villi are finger-like projections. This is seen in the lumen of small intestine. Then, to prove exoosmosis and endoosmosis, materials required: 3 beakers, petri dish, salt, dilute HCl, toilet cleaning acid, 2 equal sized raw eggs, cotton to wipe, a strip of paper for measurement, and 1 tablespoon. So children, let's see the activity in the form of video.
Now children, I have dissolved 2 eggs, raw eggs in the cleaning acid for the experiment. Children, I have removed the deshelled eggs from the cleaning acid. You can see they are very soft. Now wipe it with cloth. Now we need to measure the circumference of the egg. Pause. Here. Okay children. This is the deshelled egg which I am putting in salt solution, and this is water. Now let's make salt solution. Children, this is salt. Now I am pouring water in other beaker. So children, in one beaker I made a salt solution, in other beaker it is filled with water. Now place the eggs. Stir it well until it dissolves. Now I am placing the deshelled egg in the salt solution and another deshelled egg in the water. Leave this experiment for 3 to 4 hours. Now children, I have removed the deshelled egg from the water. This was the mark which I marked before dissolving in water. Now let's see the circumference. It is more. You can see the difference. Yes children. The deshelled egg which was placed in water swelled. That means there is a movement of water molecules inside the deshelled egg. Now children, I have removed the deshelled egg in salt solution, wiped it in cloth, and initially during the first activity the shape of the egg was measured to this limit. Now let's measure. See, the size of the deshelled egg is decreased, isn't it? Because I have placed in salt solution.
So children, you have seen when I place the deshelled egg in the salt solution and another deshelled egg in the water. The deshelled egg which I placed in the salt solution shrinked. The deshelled egg which I placed in the water swelled. Shrinked, swelled. Why did it shrink? Because I placed in the salt solution. Why did it swell? Because I placed in the water. So let's see the result. The shrinking of egg placed in the salt water is due to the exoosmosis. It means in which the water molecules leave the cell. So the swelling of egg placed in the tap water is due to the endoosmosis, in which the water molecule enter the cell. Children, endo means inside, exo means outside. So the water is leaving the cell. So exoosmosis. So here the water is entering the cell. So it is endoosmosis. So preparation of a semi-permeable membrane. For that we need 2 egg membranes, 3 beakers, sugar, water, thread, measuring jar and disposable syringe. Children, let's see the activity in the form of video.
Now children, I have dissolved 2 raw eggs in cleaning acid for the preparation of semi-permeable membrane. Children, you can see Slowly the egg is dissolving. You can see the broth also. Now pierce the egg membrane and drain out the contents. This is the egg membrane. I need to clean it well. Now in this membrane I will fill tap water. Now children, the egg membrane is filled with fresh water. Tie this membrane with thread. Now I have placed this egg membrane filled with tap water. I have made sugar solution. Now beaker has sugar solution and the membrane has fresh water. Now children, this egg membrane is filled with the sugar solution. With the help of syringe I have filled the membrane with the sugar solution. Now I took beaker which is full of fresh water. Now children, you can see now in this the beaker is filled with sugar solution and the membrane is filled with the fresh water. In this the beaker is filled with the fresh water and the membrane is filled with the sugar solution. Leave this experiment for overnight. Children, we have filled sugar solution in the egg membrane and beaker with the fresh water. You can see there is increase in the size of the egg membrane. That means water from the beaker has entered into the egg membrane, that is sugar solution. So there is a movement of water molecules from lower concentration to higher concentration, right? Here in this membrane we have filled water and beaker has sugar solution. You can clearly see the water in the egg membrane drained out towards the sugar solution. That means the size of the egg membrane is decreased. That means the movement of water molecules from egg membrane to the beaker. Beaker has sugar solution. So again osmosis process has taken place. That means the movement of water molecules from the lower concentration to the higher concentration through a semi-permeable membrane.
So children, by the above activities we have seen the preparation of semi-permeable membrane. Right? In one membrane I have filled sugar solution and other membrane I have filled water. Whatever the conditions may be, the water always move towards the sugar solution. Means children, the water has the lower concentration, sugar solution is the higher concentration. So the movement of water molecules from lower concentration to higher concentration through a semi-permeable membrane is called as osmosis, right? So let's see the importance of osmosis in living organisms. So water enters the roots through osmosis. Water moves between the cells through osmosis. It helps in opening and closing of stomata. Next, it brings about the movement of water and minerals in certain plants. In our body, waste materials are filtered from the blood. In our body, useful materials are absorbed along with the water through osmosis. So children, in the above activities, we have seen the process of osmosis. So let's see the process of diffusion also. So let's begin. So to observe the process of diffusion of coffee powder and potassium permanganate in water, let's see this osmosis or diffusion. So let's see the diffusion process in the form of video.
Children, let's do an activity to know the process of diffusion. Coffee powder. Now let's make a small bowl of coffee powder. Now children, I am making a ball of coffee powder, putting it in the water. You can see the molecules are distributing uniformly across the solution and uniformly distributed in the bowl of water, isn't it? Now children, this is potassium permanganate. I am placing potassium permanganate in center of the bowl. Now I am adding water. You can see the uniform distribution of potassium permanganate solution. This is the process of diffusion.
So The same way, the movement of molecules from higher concentration to lower concentration is diffusion. So, what is the difference between osmosis and diffusion? So, diffusion. What is the diffusion? From higher concentration to lower concentration, the molecules are spreading. What about the osmosis? Yes, in the process of osmosis, the movement of molecules from lower concentration to higher concentration. But here children, in the process of osmosis, there is a membrane, that is semi-permeable membrane, right? Higher concentration to lower concentration, there is a movement of molecules, right? So, reverse osmosis. Osmosis we have no. Freely the movement of molecules from lower concentration to higher concentration through semi-permeable membrane. Here in reverse osmosis children, the same thing is applied, but there is applied pressure, that too on the salt water. The salt water is allowed to pass through the semi-permeable membrane. So, the direction of water flow takes place and the salt will remain. So, this is called as reverse osmosis. All the purifiers which we use in the home are based on the reverse osmosis. You know, I have told you the nature of plasma membrane, that it filters the waste materials also. So, in the process of dialysis, I will give you a small introduction of it. In the process of dialysis, why do we need dialysis? When the patient's kidney has failed to perform the function of excretion, we perform dialysis. What is dialysis? Removal of waste materials from the blood. So, the blood from the main artery is taken and added with the heparin. Now, what is heparin? Anticoagulant. Why are we adding the heparin? Because children, when the blood vessel is cut, the blood oozes out. Upon 3 to 6 minutes, it clots, isn't it? It clots. So, when the blood vessel is exposed outside, it clots. That's the reason they are mixing the heparin. So, it is mixed with the heparin and it is allowed to pass through the tubes. So, this box is filled with the dialyzing solution. So, this is acting like a semi-permeable membrane. So, it is separating the waste materials from the blood. So, waste materials are separated and the blood is again taken out and mixed with the anti-heparin and pumped to the vein. So, this is the process of dialysis. So, what we have learned children? Osmosis, the importance of plasma membrane, preparation of plasma membrane, that is semi-permeable membrane, and the diffusion. So, improve your learning. What do you mean by permeability of membrane? Explain with suitable example. If the dried vegetables are kept in water, they become fresh. What is the reason? Draw the flow chart showing the different stages in doing the experiment with egg, how diffusion is useful in the everyday life. For better understanding of the concept, solve the worksheet. Please visit the official website of the SCRT. So, children, helpful to build the concept of osmosis and diffusion. Thank you.